Now I will request Samruddhi madam to present. She is from Bio Innovations. Is she reported? Please come, madam. She has some new technologies and new product to share.
Good afternoon, everyone. My name is Samruddhi Patil. I'm application specialist from Bio Innovations. So Bio Innovation is, let me tell you about my company first, that Bio Innovation has been in the field for 15 years of life sciences. And we work closely with customers to help them address complex laboratory demands. Not only microbiology, but we have diversified our solutions in the field of genomics, proteomics, and as well as toxicology. So today I'll be talking about the MALDI-TOF technology again by Bruker, as Bruker is the pioneer of the MALDI-TOF technology. So Bruker has latest system that is MALDI Biotyper Cirrus system. I will talk about the technology part as well as the application parts here. So basically, hello. So basically this Bruker system is very well advanced and up-to-date system. This is the first system that is CE IVD approved. Also, the identification is, most of the systems nowadays provide identification that is only protein-based. But this is the only system that gives lipid-based identification. This has positive and negative ion modes. So we'll talk about this in detail further in the further slides. We have vast database that gives you the most accurate identification. It has dual laser, that is primary laser, which gives you 7 years warranty, and secondary laser, that is IR laser, which cleans the detector. It has proprietary modules also that are IVD approved, such as Sepsityper and Star BL. We'll talk about it in detail. So basically, multi-biotyper is better, faster, and cost-effective. This system detects gram-positive bacteria, gram-negative bacteria, aerobes, anaerobes, filamentous fungi, yeast, MTB bacteria, NTMs also, by choosing the unique proteomic fingerprinting. So it has a very simple protocol. So only the isolated colony from Petri plate has to be picked up with a toothpick, place it on the target plate. HCCA matrix has to be applied, and it can be loaded into the system. The system will read the actually our previous presentation have already shown how the MALDI technology works, so I will not go into the depth. So it is again matrix-assisted laser desorption ionization time-of-flight technology, and the spectra is generated. So now talking about the spectrum, this is how the spectra looks, and this is how the spectra looks. Once the spectra is generated, the pre-installed library then matched with the spectra, and it gives you the result, what organism exactly it is. Now, once the spectra is generated, it gives you the score. The score should be above 2 points. So if the score is above 2, it is of high confidence. If it is below 2, 1.7 to 2, then it is of low confidence. It could be because the culture is, it is not processed properly, or the colonies are old, etc. And if it is below 1.7, then it is not acceptable to report. We have to repeat the sample, or it is possible that it could be a novel organism. So talking about the spectral library is the heart of every MALDI-TOF technology. And as the previous presentation had shown, that Bruker's MALDI-TOF technology, the number of species at Bruker MALDI-TOF technology, those were the older numbers. So in the recent advancement, you can see that there are 5,000 species. You can see here that in 2010, we only had 1,850 species in the database. And as the years have gone, in 2020-21, we have 3,800 species. And in 2022 updated library version, we have 5,000 species. The heart of the MALDI-TOF is basically the number of species. Unless you have those many species in the database, the accurate diagnosis is not possible. Accurate detection of the microorganism is not at all possible. So we have a specialized database for mycobacterium. There are 208 species, clinically known important species of mycobacteria, out of which 182 species can be identified with this MALDI-TOF technology. That comes up to 97% of all the mycobacteria. In this, it does not even only the Mycobacterium tubercle, all the NTMs are also covered here. Next is filamentous fungi. We all know that clinicians face a lot of problem identifying this filamentous fungi. Doing it with microscope is a big task, and it varies person to person. So for filamentous fungi, we have 247 species, and approximately more than 10,000 strains for filamentous fungi. And this can, because of this, the filamentous fungi can be identified with high confidence in the Bruker's MALDI-TOF system. So now let's talk about why sepsis is important. In sepsis, as we all know, every minute counts. Earlier, I was working in Hinduja Hospital on ICMR's project, wherein I worked on AMR surveillance program. And there, while working on that program, we designed antibiograms, we made antibiogram, and we made it in such a way that clinician could start empirical treatment appropriately for the critical patients. Now, since we have to start the empirical treatment accurately, we need to know the organism and its resistant pattern as soon as possible. As the time increases, the mortality rate also increases. So, to come up with that solution, Bruker has this IVD approved sepsidiaper kit. Now, this sepsidiaper kit has a protocol of say 5 to 6 minutes. And once your blood culture bottle is FAC positive, immediately we can withdraw the blood, and with this 5 to 6 minutes protocol, the culture can be identified, and we can inform the clinicians what culture it is, what organism it is, and the escalation or de-escalation of the antibiotics can be done. In addition to this, as I told you about AMR resistant pattern is also very, very important. And in ICUs, most of the time we see that the patients come up with, critical patients, especially cancer patients or dialysis patients, come up with CRE patients. They are carbapenem resistance or cephalosporin resistant, and also sometimes colistin resistance also. So for that, we have Star Carba and Star Cepha kit available, which are also IVD approved kits. With this sepsidiaper kit, simultaneously, this protocol can be run, which is only 60 minutes protocol. And we can inform the clinicians at least whether the carbapenem resistance, the organism is carbapenem resistant or susceptible, or cephalosporin resistant or susceptible. Moving on next, I told you that our MALDI has negative and positive ion mode, that we not only identify proteins, but we also identify lipids. So small proteins can identify and can give you accurate bacterial identification. But with lipids, we can discriminate the closely related species. Also, the colistin identification, chlorine resistant identification can also be done. This is the exact example, like if in every other routine MALDI-TOF technology will get the result as M. abscessus complex. But with lipid ion identification, we can identify all the species here. Wait, just a second, here. We can identify, discriminate between all the species. So how this is important? Also, one of the major problem that clinicians face, which are already using MALDI-TOF technology, the MALDI-TOF with only protein ion source, like only positive ion source, the discrimination between E. coli and Shigella is very difficult. Okay, MALDI generally fails to identify between E. coli and Shigella. It misidentifies. With negative ion mode, that is with lipid profiling, we can easily and accurately identify between E. coli and Shigella. And this is how we can save the time that in day one, once your culture, once you receive the sample, that is day 0, and within day 1, you can identify the organism. So your second and third day is saved here immediately. So to sum up, these are the 10 reasons why to choose MALDI Biotyper series. It is a benchtop model with dual ion source. It has a primary laser of 200 hertz, that is nitrogen laser, which has capability of detecting positive and negative ions. Colistin resistant capability is also there with negative ion mode. Okay, so colistin resistance can be detected with negative ion only because colistin resistance is because of the lipid A molecule. So that is why the lipid profiling is very, very important to detect the colistin resistance. Secondary laser is 60 hertz, that is IR laser. So this basically the detector of the Malditov, the detector present in Malditov, that has to be cleaned. The ions are accumulated on the detector. So with this IR laser, the detector has the self-cleaning capability. This makes the system very low maintenance and user-friendly. Sepsityper modules also gives you the edge of identifying the direct pathogen from the positive blood culture. The AMR detection can also be done. Mycology suits gives you a filamentous fungi identification, yeast and molds, most accurate identification. We have reusable as well as disposable plates. So using reusable plates saves your cost. The cost per test is not more than, I would say, 40 rupees. Also, since these are reusable plates, the biomedical waste is also saved. It has a flexibility to run the sample multiple times. This is the only Bruker's features only. Like if you want to check the bacteria, the spot again, you want to check whether the spot, you want to analyze the spot manually, then that can be done with Bruker system only. Only Bruker gives you the MicrobeNet module, that is the CDC's endorsed library. So CDC has their different library of spectra that is available to the Bruker's customer, which gives you the most accurate identification. As I told you, library is the heart of every Malditov technology. So this is how the this system is, stand out and is unique of its own. And yeah, the capability, this has the capability to do serum protein analysis also. The proteins can be identified, unknown proteins can be identified with this system also. So with this, I will sum up and thank you so much for listening to me.
Rudy, for your informative talk. Now I would like to invite Dr. Vidya Arjun Wadkar to felicitate Ms. Samruddhi. Taking this session ahead, I would like to invite Dr. Girish Mahajan, Senior Vice President at High Media Laboratories. Dr. Girish Mahajan has more than 18 years of experience in pharma, research and development, quality assurance, microbiology operations, fermentation, metabolite extract management, microbial genome mining. He has published more than 150 papers, book chapters, media writings and catalogs of bioactive compounds and filed over more than 20 patents. Now, Dr. Girish Mahajan will enlighten us on the services provided by High Media Laboratories, mainly microbial ID today, that is Malditov. Welcome, sir.
I'm the last batsman, and I'm the intermediate between your lunchtime and ongoing session. So I'll definitely try to keep it as brief as possible. So first of all, thank you very much, the Maharashtra MicroCon conference organizer and Karyakarta sir, for inviting me and creating a forum to present scientific part of microbial ID. So again, let me take you ahead a little bit. So High Media as such, all of you are very familiar, and it has been almost 48 years. High Media, the indigenous Indian company, started with microbiology products and now diversified into molecular biology as well as cell biology, hydroponics, means agro-related nutrients. So today, this is the new headquarters of High Media at Thane, near Mumbai. And also we have the offices at Germany and USS and CGMB factories in 4 different cities near Mumbai. So this is just a profile portfolio 7,000 products and diversification in bioscience field. And of course, today we have DSR recognized R&D lab since last 25 years. And we have various quality certifications and product registrations in US FDA also. And again, since last 3 years, we have been an authorized partner of all ATCC products to deliver our patterns in India and the services along with the products. So today, basically, my earlier colleagues have already set a good stage for Malditov. And also, I will be talking more of the Malditov. Since there are many students, I thought it is better to take the principle behind the Malditov. Technology, we all of us know. So let's go a little bit about the science behind it and the basic fundamentals, what exactly Malditov does. So there are, if I consider the methods used for identification of the microbes, so there are indirect methods as well as direct methods. Conventional methods, isolation of culture, microorganism, and determination of their various phenotypic characters, which involves biochemicals also. And culture independent method, which involves starting from microscopic as well as Malditov comes under that. So this is a small chart which tells us about the different ID microbiological methods which are used. Use of traditional media followed by microscopic techniques, biochemical techniques, and we have molecular therapeutics which covers both 6-ness, 8-ness RNA as well as the Malditov. So the technology of identification of microbe has been evolved tremendously. And basically, there are two needs. One, assurance, the confidence level, and secondly, the fast, the speed at which the clinicians need to identify the microbes. So perhaps these two needs has helped the scientists to evolve the faster techniques, and Malditov perhaps is one of it today. So I'll not repeat this again. Now basically, if I consider the earlier classical techniques like visual, smell, taste of the smell of the colonies, as well as use of monoclonal antibodies, PCR methods, RFLP, rapid metabolic capacities, lipid profile, sometimes antimicrobial resistance pattern of the culture. So all these techniques have some limitations. So obviously some of the disadvantages which we found, key disadvantages, limit the idea of the microbial species. Complicated processes, it needs a very skilled manpower, time costly, time-wise it is very costly, and very expensive with respect to cost. Per test costs are sometimes very high. Now compared to that, MALDI, matrix-assisted laser desorption ionization, and TOF, time of flight of the mass spectroscopy, in which we actually find out the time of flight of the proteins which are flying inside the vacuum tube. As such, Malditov has been compared to recent technology of the 1980s, and the first Malditov for microbial ID has come up in 1988 officially by some of the earlier pioneering companies. So identification is possible for aerobes, anaerobes, mycobacteria, mycoplasma, fungi, and the biggest USP of Malditov as a technology is fastest, accurate, cost-effective, very constantly updating database, and greatly improves the efficiency of microbial identification. Now the question is, what is so unique? Without any reaction, without any biochemical, without any incubation, how do you get directly identity of the microbes? So it is like scanning. So all microbes, fungi, bacteria, they have ribosomes. And there are certain housekeeping proteins in each of these. And these ribosomal proteins actually comprise about 60 to 70% of the total dry weight of the bacteria and the fungi. So these have special proteins which are very unique, the signature molecules of those genus and the species. And these are the one which Malditov technology had to bet upon. So by Malditov, we try to identify the molecular weight of this, and complete molecular profiling is done with this. And this profile of molecular weight, and especially for Malditov, we choose 2,000 to 20,000 Dalton molecular weight, because this range of molecular weight of proteins is very unique to the bacteria and the fungi. So these are the picture of ribosomes. So as such, again, I will skip this slide, some of the things already told. But principle-wise, I can say what is done exactly. So usually, on a target slide, we have to make a thin film of bacteria or the fungi. Then you add the matrix. Now, this matrix is very unique. This matrix co-crystallizes the proteins which are released from the bacteria and the fungi. And once they are co-crystallized, the laser beam, which is energy beam, it helps the protein molecules to fly. And based on the molecular weight, they fly up. So this schematics will tell you. So here on the left side, laser beam is bombarded on the sample. And these molecular proteins are moving up and reaching to the mass detector at the top. Lowest molecular weight will of course come first. And based on the type of molecular weights, a profile is generated. So this profile is the signature of every bacteria. And this profile is compared with the database inside the system. And immediately the report comes. Now, when you choose MALDI-TOF as the technology for identification, we have to have some criteria in the mind, apart from understanding the principle. Number one is, how big is a database? My earlier colleague told the significance of this, but again I'm going to reattire. Database is very, very crucial in this. If the database is very low, up to species level, so obviously it's of no use. In most of the reports, you will say peak not identified. So how big is a database? That is a criteria clinician should ask when somebody comes for MALDI-TOF sealing. Number two, how many species are there in the database? Many times it's confusing. Some people show strains, big, genus, big, but what is important is from clinical perspective is the species. How many laser shots can be generated? The laser shots in our MALDI-TOF, the lowest version, 400 million laser shots. And the higher version, we have 7 billion laser shots. It means, what is million billion means? It means my laser is very, very stable. We are providing a warranty of 10 years. So laser shot is very important. And capacity of the vacuum pump, which is again very crucial. The capacity of the vacuum pump will decide the strength of the vacuum created in the MALDI-TOF tube. And that will reflect to how fast the report is generated. Time of result, of course, that's in few seconds. If I insert the site, target site and say scan, it's a question of 45 seconds. The report is in front of you. So this is how the basic MALDI-TOF looks. Its model number is MS1000. So these are the most common accessories which are along with our MALDI-TOF. This is the unit on the left-hand side. And target plate is there. It also needs a 10 microliter pipette. And the rotary spinner is required for fungal growth. Because again, the success of MALDI-TOF identification also depends upon how young culture you are selecting. The culture should be in a log phase so that the amount of ribosomes is very high. And these are the reagents we are providing, ready to use reagents, no need to mix A, B, C and make a array data of it, but directly we can use the reagents. So the minimum operational steps, first transfer the colonies with a finest needle we have in the lab, and then touch the target plate, make a nice thinness smear. Again, we have to put a lump there. We have to make a thinness smear because laser is hitting that. And then add the reagent and put in the hatch of the MALDI-TOF. The report is ready. So final review by the microbiologist and give the report. And though every MALDI-TOF will give you, first top 10 or top 3, so we can decide based on the score. Now, in our system, the interpretation is very simple. So we have a big range, 0 to 10. If the score which is coming on the screen when you do the scanning, if it is 9.5 to 10, the confidence level is very high up to species as well as subspecies level. If the score is 9 to 9.5, of course, the reliability and the confidence level is very high up to species level. That's why green zone. If the score is between 6 to 9, then the reliability is up to genus level. It means if I say Staphylococcus aureus, but score is 7, it means it is Staphylococcus aureus, but confidence is not aureus. It could be epidermidis or some other strains. So in that case, we have to do the extraction of the protein and rework. And majority times, if I extract the protein, it works. Because many of the bacteria, based on which medium they are growing, they are loaded with polysaccharide, polypeptide in their envelope. And these cause a hindrance in the matrix and a laser shot. So obviously, we know the protein extraction. And of course, it is 0 to 6. It's not a reliable ID. So perhaps you have to repeat the whole procedure itself, or it's a good luck for the researcher. He is handling some new species or sometime new genus. So our fast to time results as compared to the conventional ways and the PCR, accuracy similar to nucleic acid sequencing, very cost effective. And if I consider only the reagent which is required for it off, the cost per test is only 17 rupees. I'm saying only with respect to reagent. Robust, intuitive software and supporting 20 CFR Part 11, installation, qualification, operational qualification, performance, of course, these supports are given by all of us. And the database, the biggest, 5,053 species. And in fact, I have a good news from my parent company that there is further change. But at present, I would say that I have a species of 5,053, around 17,000 strains, and can identify 96 samples in less than 20 minutes. So within an hour, I can find out the idea of about 300 cultures. So this is a vertical representation of the same. So the auto of MS-1000, though we are the new players in this, but very dynamic players, because the technology which was there previously, it has been improved, advanced, and made faster and more accurate. So there are about 17 inventory patents in each circuit and each screw which is used here. So I can say again, currently, we are the world's biggest microbial database as compared to other service providers. And of course, I use the word service providers at that. Along with the Maldi, we are also providing the service of the identification. That's why I use the service providers also. So about Multitop as such, unlimited identification in theory can deal with most kinds of colonies in simple pre-treatment. When I say colony, means simply you have a well-grown plate, just pick up, touch that colony, and do the identification. So in a fraction of 2 minutes, the thing is done. Very easy to get the pentamole sensitivity and high throughput, but low cost, almost no cross-contamination. Because the target plate is designed in such a way that when you spread, there is no cross-contamination. And one more thing is that along with the regular identification, it will also help you to identify the proteins. If you have certain peptones for which you want to find the molecular weight, it will work easily. There are different metrics provided along with that. So this is all I wanted to share with you today. Thank you very much for your kind attention.
Thank you so much, Dr. Girish Mahajan, for your valuable talk on microbial ID today. Now I would like to request Dr. Meghna Palewar to felicitate Dr. Girish Mahajan, sir. I would like to invite and thank Dr. Sharad Agarkhedkar, sir, as an MMC observer for our conference. So please come on stage. I would like to invite Dr. Vaishali Vabre, madam, and Dr. Karyakarte, sir, to felicitate Dr. Sharad Agarkhedkar. And Dr. Karyakarte, sir. Now I would like to invite and thank Dr. Meera Modak, madam, and Dr. Swati, madam, for conducting the wonderful PG quiz. It was indeed very informative and interesting. Please, Meera ma'am and Swati ma'am, come on stage. to felicitate Dr. Meera Modak, madam. Aparna Joshi, madam, to felicitate Dr. Swati, madam. Thank you. I would request Dr. Swati, ma'am, to please display the answer of PG quiz. Yeah. Now we will begin the valedictory function. I would like to request Dr. Sushma Pednekar, ma'am, to start the valedictory function.
Request all PGs to assemble in the MJ auditorium. We'll be shortly displaying the answers of the PG quiz. First MTFI, I think I would not repeat the questions again. The PGs can go through the answers. So it was prevalent in Bengal, pathogenic strains on Vagatsuma agar, and they have common H antigen, and of course they belong to Vibrionaceae family. Regarding Niglavia, these were the answers. The acquired infection by swimming in freshwater lakes, ponds or swimming pools. Regarding question 3, they can survive at 4 degrees Celsius, and the hitch was Palcum agar. It does not contain maltose, but it contains mannitol. Okay, and the last was true. This was the answer for the MTFI-4. Okay, so hair perforation test is positive in Microscopum canis, and color changes due to alkali metabolites. We all know immediate hypersensitivity reaction, it is IgE dependent, not IgG, and adverse reaction is type 3. These are the answers to the MCQs. If PGs are attentive, they can go through. It is nothing but the toxic shock syndrome caused by tampon, nasal tampon, and it is, so that's why a toxin, that is superantigen TSST, that is the culprit. Typhoid dot is outer membrane protein antigen is used. Burkold West pseudomyelai, it is called as Vietnam time bomb disease. I would like to tell few words about this. Why it is called as time bomb? Because the soldiers who acquired the infection during the 1947-48 war, they did not acquire the infection immediately. After there was incubation period of 2 to 3 years, and later on they all developed infection. So the bomb was about to blast. So Burkold-West pseudomyelai is the answer. Culex vishuni for Japanese bean encephalitis. Of course, this is a tuberculin test, delayed type of hypersensitivity. So cytokine production by CD4 lymphocytes is the process for induration. Among the Hasse group, due to the human bite infection, Eikenella cavodens is the infection bacteria associated with the human bites. For relapsing fever, direct detection of spirochetes in body fluid is the answer. For Helicobacter pylori, curved or spiral bacteria should be seen in biopsy after silver staining. Antemortem diagnosis of rabies, corneal impression smear for immunofluorescence, and receptor binding is observed in T cells. This was the answers for mix and match. Majority of the people, PGs, they failed to answer it correctly. So these are the answers. For mix and match 2, it was the answer. You can click it and later on see. This was a Romaner's sign because of the organism, that is a parasite, that is Trypanosoma cruzi, and the cells and tissues affected are myocardium, skeletal muscle, and neurological cells. This was the tissue cyst of Toxoplasma, and they are nothing but the slowly multiplying bradyzoids, and they are found in muscle and other tissues including brain. This is nothing but the chancroid, that is soft chancre, caused by Haemophilus ducreyi, and the typical morphologic appearance is pleomorphic gram-negative cocobacilli, school of fish, or railroad track appearance. This is visual for, they are nothing but the fried egg appearance colonies of Mycoplasma pneumoniae. And liquid medium used to is PPLO broth. What is PPLO broth? What is long form of PPLO? Pleuropulmonary-like organism. So that was the initial, that is called as PPLO-like organism. It was previously called as Mycoplasma pneumoniae. So that name of the broth is PPLO broth. Nobody could identify it correctly. Actually, of course, the answer to the question was Syncephalastrum racemosum. Why? Because you could see in the picture here, there amongst the mucormycosis family, because you can see here rhizoids at the base. So it was, and secondly, you can see the broad accepted ribbon-like hyphae, and this sporangiophore and conidiation, which is seen, is a flower-like pattern. So that is typical of the Syncephalastrum racemosum. So we last year only we have faced the second pandemic of mucormycosis. So I think many of the people have got these isolates along with Rhizopus, mucor, and Rhizomica. Syncephalastrum was the etiological agent. So diseases caused are rhinocerebral mucormycosis and invasive pulmonary mucormycosis. So these were the answers to the visual round. In clinical one, so this is nothing but a case of inclusion conjunctivitis. So caused by, so the probable organism was, there was a clue given, Giemsa stain showing cytoplasmic inclusions. So the probable organism was Chlamydia trachomatis, and the name of the including body was, they are called as HP bodies, that is Halbersteder Provazek including bodies. So they are the diagnostic for diagnosing of trachoma. So I think this was answered correctly thanks to the work we did, all the microbiologists, work done by all people last in the pandemic. So screening genes are S, E, and N, and for confirmatory genes are ORF1AB and RDRP, so, and N2. So these were the confirmatory genes. But many of the people have failed to answer the name of the prognostic markers. So that was IL-6, D-dimer, and ferritin. So increased levels of this, they are called, they are prognostic markers for COVID illness. In this question, it was a pulmonary histoplasmosis. So X-ray showing infiltrates, biopsy of the lymph node was positive, and on SDA agar there was mycelial growth, and the clue was spevical spores with tubercle. That is the typical morphology of histoplasma capsulatum. And how you convert mycelial form to yeast form is by growth at 37 or by growing it on blood agar. This question, the child presented with the CNS symptom, that is seizures, irritability, and fever, and poor feeding, and there was history of prolonged rupture of the membrane. So the organism, so this diagnosis is basically early onset meningitis, and etiological agent is Streptococcus agalactiae, that is group B Streptococcus. And what are the two tests used are Hippurate hydrolysis test and CAM test used for diagnosis of group B Streptococcus. Clinical 5. So this was an outbreak of a tick-borne disease in the military camp, and blood smear showed ceftiofin appearance. So that was the clue word, ceftiofin appearance inside the RBCs. So the pathogen was Babesia. Okay, one of the tick-borne pathogens, and the ticks responsible are Ixodes, Buphilis, and Dermacental. Regarding relationship analysis round, So the daptomycin failed to act in the lung infection. So both assertion and reasoning are true, and assertion is the correct explanation of the reason is the correct explanation of the assertion, because daptomycin is a peptide antibiotic, but it is inactivated by surfactant, so it cannot be used, or it is failed to act like in the ventilator associated pneumonias and lung infections. Relationship analysis 2, antigenic drift account for major epidemic or pandemic, and that was the reason given. But here both the assertion and reason are false. In this question, Yersinia enterocolitica produces different reaction on TSI and KIA. Here assertion is true. It produces different reaction on TSI and KIA, but reasoning is false. Why? Because it ferments only, that's Yersinia enterocolitica, only ferments sucrose but not the lactose. And TSI contains additional sucrose, but which is not a component of Kigler iron agar. That's why it failed to show the same reaction on the KIA medium. Here, regarding this varicella, both the assertion and reason are true, and reason was the correct explanation of the assertion. So this was the answer. And last question, that is presence of capsular polysaccharide is an important prevalence mechanism for Streptococcus pneumoniae. So assertion is true, but reasoning, that is capsular polysaccharide acts by inhibiting inflammatory response by activating CD4 cells, but that is wrong because it is complement mediated process. So that was the answer. Okay, so I think with this we come to the end. Thank you all for giving me the opportunity to conduct the quiz. I thank organizers and thank you all of you for the same. Thank you.
Much delay, will proceed for the valedictory function. I request Dr. Neena Nagdev, President IMM Maharashtra Chapter, Secretary Dr. Anuradha Deshmukh and Treasurer Dr. Rajendra Surpam to come on the dais, please. Now I invite newly President-elect IMM Maharashtra Chapter, Dr. Rajesh Karyakarte, Secretary Dr. Smita Deshpande, and Treasurer Dr. Vidya Arjun Wadkar to join the stage. Thank you all. Now we'll proceed for the most awaited session of the conference with full of excitement for all of us, that is the valedictory function. All the presenters of oral and poster sessions are the winners. In this conference, we received total 43 posters and 59 oral papers. All the papers were well presented and it was challenging for our judges to decide the winners. Now I call upon Dr. Sandhya Kulkarni as a representative to brief about the oral presentations. Dr. Sandhya Kulkarni, Madam.
Thank you. I'm Dr. Sandhya Kulkarni, Professor and Head at Mimar Medical College, Talegaon. I just one session of oral paper, and actually, as we all know, we are in teaching. So because of the mandate from MCI and NMC, the PG students, they are supposed to present their research work. It's not only the thesis, but other also. And day by day, because of this, the research quality is improving. And really, I enjoyed the session. Thank you, organizers, for giving me this opportunity. Thank you.
Now I request Dr. Vijay Kulkarni, Associate Professor from Kolhapur Medical College, to express his views on poster presentations.
Thank you. First of all, I would like to congratulate respected Karyakarte Sir, Deshpande Madam, Arjun Wadkar Madam, and entire microbiology staff for successful conduct of MahaMicroCon-2022. It was a nice academic phase, especially the sequencing and all the stuff, and also the food and everything was very good. Thank you, sir. I along with Dr. Mule from Navle Medical College, both were the judges for poster presentation yesterday. We went through the posters, they were very good. The scientific content was also very good. Actually, it was difficult for us to choose the winners. Among that, there were ties in many of the papers, and so again, we had to go again and again for that to choose the winners from them. So, I would like to congratulate the winners and all the participants of the poster presentation, and I would like to thank the organizers for giving us the opportunity to judge the session. Thank you very much.
We had a very fruitful GBM yesterday, especially the financial point of view. We are happy to announce that this conference, many new awards have been introduced. Total 8 new awards will be sponsored by various eminent microbiologists of our state. Principal amount of rupees 50,000 will be deposited by each sponsorer in an IMM account of a chapter conference to give away the prizes from the next chapter conference. So big round of applause. The prizes, the awards will be as follows. First is sponsored by Dr. Rajesh Karyakarte, Organizing Secretary, Professor and Head, Department of Microbiology, Deputy Dean, BJGMC, Pune, in the name of his father, late Dr. Prabhakar Karyakarte, for the best paper in parasitology. Next is by Dr. Mrs. MP Bansal, ex-professor and head microbiology, GMC Aurangabad and MGM Medical College, for best paper in molecular biology. Then Dr. Pratibha Narang will be sponsoring the next award. She is ex-professor and head microbiology and director, Sevagram Medical College, for the best paper in mycobacteria. Dr. Abhay Chaudhary, ex-professor and head microbiology, Grant Medical College, ex-director of Hafkin Institute Medical and currently working as professor and head microbiology, D.Y. Patil Medical College, Mumbai, for the best paper in hospital infection control. Dr. Vilas Jagirdar, ex-professor and head microbiology and ex-dean at VMGMC Solapur. Sir will be it will be a best paper in virology. Dr. Raghoji will be sponsoring next award in the name of Dr. Sen Gupta as been best paper in immunology. Dr. Rajendra Surpam, professor and head microbiology, GMC Chandrapur, best poster in microbiology. Dr. Renu Bharadwaj will be sponsoring the next award. Madam is ex-professor and head microbiology, ex-dean BJGMC Pune, in a memory of late Dr. B.N. Joshi. He was an ex-professor and of microbiology, BJGMC Pune. Joshi sir was a pioneer to establish the microbiology department in BJ when it was separated from the pathology as a separate subject. We are happy to announce that B.N. Joshi Award will be given to the best outstanding paper of the conference, Maharashtra chapter conference, from this conference itself. Now I have a list of all the winners. So without much delay, I will declare the winners. So we'll first begin with the case report. The second prize winner of the case report is Dr. Mayuri from KM Mumbai. She has presented poster titled Subcutaneous PO Hypomycosis by an emerging Rhytid rufulum species. I request Dr. Mayuri to come on the dais. I request Dr. Neena Nagdev ma'am to give away the awards. Congratulations, Dr. Mayuri. Okay. Now the first prize case report goes to Dr. Akash Dhumbare from GMC Nagpur for sporobolomyces salmonellar infection of the upper respiratory tract. Congratulations, Dr. Akash. Thank you, ma'am. Now I request Dr. Anuradha Deshmukh to give away the awards. So now we'll move to the poster presentations. The third prize winner is Dr. Hira Padekar from BVP Loni. She has presented SSI in tertiary care hospital. She has presented a mucormycosis colon R experience. Dr. Hira Padekar. Is she there? Anyone from Loni, from her college institute, can take the award on her behalf? Okay, since no one is there. Okay, the second prize for the best poster goes to Dr. Pratibha S. Mane from GMC Nanded on study on bacteriological profile antibiogram of SSI in a tertiary care hospital. Okay, Dr. Sanjay More will volunteer to take the prize. Congratulations, sir. Yes, sir. And the first prize, the poster prize goes to Dr. Sweetie Singh from BJGMC Pune for assessment of Lidona defloxacin activity in a gram-positive cocci with special reference to MRSA. Dr. Sweetie Singh, congratulations. Thank you, ma'am. Thank you, Deshmukh, ma'am. Now I call upon Surpam sir to give away the next awards. Next, we are going to give the awards for the best paper in bacteriology. So the third prize goes to Dr. Asmita Gopal, GMC Akola, for microbiological profile of infective keratitis in a tertiary care hospital. Dr. Asmita Gopal, anyone from Akola? Yes, sir. Yes, Dr. Ambore sir is receiving on her behalf. Second prize winner for bacteriology is Dr. Kalpana Suryavanshi from Cooper Hospital, Mumbai. She's not there. So first prize in bacteriology oral paper presentation goes to Dr. Thinmozi, titled Emerging Resistant to Cefixime in Neisseria gonorrhea in Erogenal Center for STD in Central India. Congratulations, Dr. Thinmozi. Thank you, sir. Surpam, sir. Next, I call upon Dr. Vidya Arjunwadkar, madam, to give away the awards. So, the best paper in serology goes to Dr. Mazge from LTMS MC Sion Hospital, prospective study to compare the results of FIR test with the gold standard ELISA test in dengue patients admitted in a tertiary care hospital. Dr. Mazge, congratulations. Now moving to the next best oral paper in virology, it goes to Dr. Ranjana Thate from KEM Mumbai, role of CD4 count estimation in the era of HIV viral load among PL-HIV. Dr. Ranjana Thate, congratulations. Thank you, Vidya ma'am, for doing the honors. Thank you, ma'am. Now I call upon Dr. Smita Deshpande to give away the awards. So the best paper in mycology oral presentation goes to Dr. Ashwini Bhosle, candida isolated from the oral lesion of the patient having habit of substance abuse. Congratulations, Dr. Ashwini. In this conference, because of we had a COVID pandemic, we have decided one more section for the awards, that is best paper in SARS-CoV-2 infection. So this winner of the second prize is Dr. Sweetie Singh from BJGMC Pune. Her paper was performance of different clinical samples in the diagnosis of neonatal infections of SARS-CoV-2. Congratulations, Dr. Sweetie. And the first prize goes to Dr. Adhunika Singh from Sion Hospital, Mumbai, titled Comparison of Rapid Antigen Test and CBNAT in COVID-19 Patients. Congratulations, Dr. Adhunika. Okay, you're taking on. Thank you, madam. Now I call upon Dr. Rajesh Karyakarte, so, to give away the awards. So our again newly introduced section for awards is on sequencing. So the second winner is, yes, Dr. Hindol Maithe from Sevagram, MGIMS from Sevagram. Her paper was on whole genome sequencing of MTB complex from a tertiary care hospital, Avardha district, Maharashtra. Congratulations, Dr. Hindol. And the first prize goes to Dr. Sushma Yanamandra from BJGMC Pune. Her paper was on clinical characteristics of SARS-CoV-2 Omicron cases attending a tertiary care hospital. Congratulations, Dr. Sushma. So today morning we had conducted a PG postgraduate quiz with lot of attendance today. So I'll go for the declaration of the winners. So the second prize goes to Anna Fernandez. Anna Fernandez. And the first prize winner of PG quiz is Dr. Kartika J from AIMS, Raipur. Congratulations from Raipur. There was a prize for best paper in mycobacteria. It goes to Dr. Deeptanu Mazumdar from GMC Nagpur, a cross-sectional study on a severe prevalence of toxoplasmosis in TB patients in tertiary care hospital, Central India. Dr. Deeptanu, congratulations. Okay, now the most awaited award of the conference, that is late PM Khare award winner. So the winner of this award, yes, I request Dr. Neena Nagdev madam also to join with Dr. Karyakarte sir. So the winner of late PM Khare award is Dr. Rashmita Das from BJGMC Pune for S gene target failure in SARS-CoV-2. Congratulations, Dr. Rashmita.